Yes, my mission is to make the introductory remarks, and as you have seen in the program, so we are going to climb today and during the course, so I'm giving the introduction. So the background is first relates to what is RNA doing, why is RNA around, what is important, so important about RNA. And everything goes back to the basic principle and the principle of molecular biology that information, the transfer of information has a direction. It goes from DNA to proteins, and right from the beginning, when the genetic code was discovered, the importance of the DNA, the importance of intermediary molecules were predicted. So it came before actually they were discovered. Those were transfer RNAs, because between the DNA, the information content, and the product, the proteins, you need something. So at the beginning it was just that. Just that is quite enough. It was quite a lot. It was the translation of information, the genetic information. So from DNA, between DNA and protein, you add the RNA. So you need a transcription step to transcribe the DNA into RNA, and then the translation itself, with two key partners that you will hear about. I mean, you will hear a lot about the first one during this meeting, the ribosomes, and one which is better known, older, the transfer RNAs. But so that was the beginning. But then things evolved, and sequence were available, information was available, and the problem of the importance of the coding sequences, because the first slide referred to genetic information content in the DNA about product, and the product was the protein. And it realized that the coding part in the human genomes, while there were several steps in between, is only a very tiny part of the genome content. You see? So more than almost 99% is not coding in the strict sense of the term of the first slide. So that was a question. Maybe more than 20 years ago, we were on the prokaryotic part. This fraction is much higher, actually, but still very small. The concept of junk DNA was coined at that time. You know, what is all that about? But even if life is complicated, it's very difficult to understand why you have so much junk. It turned out now, a long story short, that it was not junk. This is very important, and especially the more complex the cells. And here we have the human genome, which belongs to higher eukaryotes, several, and then the smaller this part, and the higher the other part, and then you have other functions of the genome content, and there is other function. What is the most important one is regulation. So not only do we need to transfer the information, we need to regulate this transfer. You need to produce the protein at the right time, in the right cell, and all that needs regulation. So the regulation part is the most important one, provided the first one is accomplished perfectly. So you need both. So there you understand that it's more difficult to make the regulation. It's like in human life, and to just do the basic things. And so now we still have these flux, but here you see few other. You have these. You remember these are the two functions that were coined first, and then the protein is here. But then you have RNA is growing with respect to DNA in terms of importance, because the specificity I will come a bit later about of RNA is that RNA is doing more than just one thing. It's doing several functions. That is what is so special about RNA. You see, because RNA is just more than a receptor information content, which is a very important function, but it's more than that. It has other functions, enzymatic functions, catalytic functions, and carries several roles, you will see. But DNA, of course, is there. And then it plays a role also in controlling. You will see that DNA is not just a dull molecule, which in higher eukaryotes is packaged in chromosomes, chromatin chromosomes, complex. The structure of DNA is important, how it is relaxed, open, transcribed, is also regulated, but it's regulated by other molecules and also by its own modification. And all that is performed by proteins, which are product, and the whole scheme is interconnected. And in all this scheme, RNA is a key player, very important player. Okay. So we call that this modification epigenetic. So regulator RNAs maybe, now I think I should stop to say more important, not important, as important as the other, because every function is crucial, as you, of course. The translation part is essential. We wouldn't talk about regulation without translation, of course. So the non-coding RNA is functional RNA molecule which do not code for the proteins. Very important part. Interfering gene expressions were discovered in 1998. See, where the transfer RNA discovery was in the 50s, at his prediction. And so 98, microRNA. I mean, there was a clear acceleration of the content of information with microRNA, which degrade or interfere with the expression. These are three aspects with two new functions which are very crucial. This is what really makes RNA special. And behind that, you will see the basic nature of RNA is embedded and is important. And this course will tell you more for each of these functions. So we have here just, I'm summarizing, so I'm very brief. You understand that. So I just put some highlight key points, and then you see. Type of functional RNA where the function is controlled by Watson-Crick base pairs, classical arrangements. And this is most of DNA also, structure. And RNA where the function is controlled by the RNA architecture. This is what is very new in terms of RNA compared to DNA. DNA has also an architecture, but it's always double helix around it with nucleosomes, packaging. It's very important, not being minimized. But here we are talking about complex architecture, which is really the type of what you have in fold of proteins. And there, the non-Watson-Crick base pairs are playing an important role. So the other parts of the RNA which are interacting are very important. So there is, with RNA, a great diversity of function and architecture. They go together, of course. As you will see, we'll come back. Everything we are talking about today is RNA, RNA function. These are molecules. What is behind the functional molecules? Chemistry. So you have to know that the chemistry is crucial to understand what is going on with RNA. So here we have the genome. That's a double helix. Ribozyme, that's a revolutionary new concept that came out in the 80s, where RNA was more than just nucleic acid that would transfer or be intermediary. You would have an action, chemical action. Here you have the transfer RNA. And you see ribozyme, the very short pieces of very complex structures. It's also complex it is very diverse and complex in complex formation with other molecules and proteins. So you have a very large varieties of interactions, of recognitions. Of course, it is obvious, since the structures are so diverse, there's the interactions they made with protein partners are very diverse and very complex. You have more than just the classical helix recognition. Okay? I'm not going into that, just showing a few glances of what is important about RNA, what makes RNA so different, so special, let's say. And as I said, you know, everything we are dealing when we are talking about RNA function is chemistry. For doing good chemistry, you need the structure and the function. That is. So the structure is important. What is the base, the database on which we build upon to understand the structures of these molecules? And the most important one so far is the protein database. Protein is not, I mean, protein database includes also nucleic acids, of course. So the PDV structure, you see the number of proteins, that was things that was compiling at the end of 2008. So the numbers are just, that the proportion doesn't change very much. You see the number of proteins content, 50,000. The database, that's pretty large already. But when you have protein DNA complexes here, protein RNA, less than 1,000, mixed complex, then we are 54. And DNA, RNA, it's about the same, and DNA, RNA complexes. So here you see that we have quite a lot of information, but it's not as much. I mean, it's still on the wedge, because it is difficult to get these structures, especially with RNA, because RNA is a fragile molecule. It has this dynamic, this behavior complexes, so it's difficult to stabilize a major conformer that can be packed and form a crystal that can be tracked and studied by crystallography, for example. That's why these numbers are lower. It's not just the variety, of course. Begin here, a lot of these structures here are rather routine works, I would say. But still, I mean, the major difficulty is there. And especially when you are talking about these complexes, you have to bear in mind that you have very often transient complexes. The most important functional ones are the ones which are formed. They have a stability, but by essence in the life of the cell, they have to go apart and recombine. So the structure is dynamic. And you have an order and the flexibility. So that's why this is still a challenging process. So now, a large background, now more narrow background. Chemistry and structure of RNA, since I'm here. So what makes nucleic acid so special compared to proteins? Of course, we'll see the chemical structure, and they are negatively charged biopolymers. So are chains. This is like protein, but instead of amino acids, you have bases and the backbone, a ribose phosphate backbone. So the charge aspect of this electrolyte makes it very special. That makes special also for the interactions. It stresses the importance of the ions in the buffer, in the cell, of course, control. That's why you have ions like magnesiums, which are very important functionally and structurally. I said just to give you a glimpse, an example of where does this information come from. Most of the information I'm referring, and several our colleagues will speak about the structural information, comes from basically crystallography. What is a crystallographic structure? Is that you have the electron density here, and the electron density is interpreted in terms of model. So here you have what is called resolution. That's 1 angstrom. That's pretty high. I mean, that's the highest level. It's comparable to what you have in small molecule crystallography. And you see that you can very clearly see the nucleic acid, the base, ribose here, and you can see an ion here, water molecules, you know, everything. Just to give you a comparison, this was the 3 angstrom information in 1985, just standard, not hyper refined. You see the quality of information? This is why you have, of course, now a 3 angstrom map today would be better than that because of progress of the techniques, the data, quality of the data, quality of the phasing. But just, you know, a lot was built on data of the early 80s. Actually, the structure of transfer RNA, the first one was in 74, 10 years before. So I can tell you the first map to be interpreted for transfer RNA, if you had a community of crystallographers in front of that map, you would have had 90% of the people wondering what they were looking at. And even with the model inside, they were that was the level. So it needs a lot of skills in interpretation. For that quality of map, you don't, a computer and program does it for you. You don't need interpretation. So here is the basic unit. Phosphate, ribose, and the base. Purine, pyrimidine. Very simple. They make oligomer. What is important is this phosphodiester bond. That's where cleavage occurs, chemistry, a lot of chemistry, recognition, the charge group are there. So the chemistry around this part is very important. What makes the structure around there is the stereochemistry, that is the organization of this chain, one beyond this other. This is one nucleotide where you see you go from one phosphate to another with the ribose here, and there's from 5' to 3'. That's the direction. Nothing special. What is special about RNA, you will see, is this hydroxyl group here. Conformation of the bases first. You have the default state, which is the base there compared to the backbone, and the syn. Syn base is very rare for DNA, very rare. RNA is much, you can find more often because you have these complex structures. So most of the conformations are anti. Anti is that compared to the backbone. You see, in order to go from anti to that, you have to rotate around this bond. And you see that this conformer, if you have a pyrimidine, won't make a lot of difference in terms of stereochemistry. But what you observe is only for purines. More importantly is the ribose. That ribose gives both the essential difference with the DNA and the dynamic. Here you have a representation of the different conformational states. Here is just when you move the pucker here, what is called the pucker of the ribose ring. Here you have two extreme positions, very simple. There are a lot of intermediary ones. The C2' endo and the C3' endo. So the C2' is here, and here is the C3', which is endo, compared to the base. So pointing toward the top here. And here is the 2' pointing toward the top in the same direction. If you take this plane. See, it goes where the base is, and here is the C3'. It looks very simple. But if you do that with the sticks, now that you have computer, you've lost this possibility to do that. But if you take physically a model, a wire model, and you just flip C2', C3', you will see this type of movement. This is not imagination. It's just the different conformers that you, by moving in the continuity, the pucker of the sugar. And you see that you can move the base from here to here, the phosphate, the dynamic. So by changing that, adapting, you have a lot of flexibility. So the C3' endo is the standard conformation in RNA helices. The C2' is non-standard. It means that you don't encounter it naturally in helices, but you can encounter when you change the conformation, you have conformational change and then allows adaptation. So you pay an energy for that price, but then you have to compensate, and you compensate with interactions. By the way, conformational change is energy. It requires energy. It's always chemistry. And so you have to find a way to provide that energy. An energy gain that you get by complexation, ions, there are a lot of means, variety, which give compensation and allows this to stabilize a conformer which otherwise would be unfavorable. So you can go on. A-form helices, which is the standard helix for RNA, and B-form, which is the standard DNA helix. Just to give you a glimpse on what that shows. Oh yes, another point is the Watson-Crick base pairs are isosteric, which means that the distance here is the same for CGs and UA. That's very important concept, you see, because having that makes helices very regular. From here to here, same distance. You can change, so you can build upon that helix by changing the text on scale, you know, the sequence. You can change the sequence, but you still keep a regular structure. So this distance here, what counts is this conformation. In order to go further, the importance of the conformational changes, just give you here an idea of what is usually called when you have an helical part. This is a normal bDNA helix. And then you see you have a major groove, deep groove, and a minor groove. The minor groove is a part of the surface which is more exposed and more hydrophobic. So here you have one way to bind where you have the phosphates very close compared to the other one where it's deeper. If you have the volume, the other projection, here is the minor groove, that's the major groove. You see the size of these grooves, that's the bDNA, the B form. From the top, you see that you have a very good overlap of helices. You have no space in between, it's just tactile. And here is the same view, where you have this groove, where you have more the volume, the CPK volume. And you see the accessibility here is very great, that's a minor groove. And now you go with the RNA type, or the aDNA or RNA is the same in terms of helix. You just change the pucker. And you see the compensation here, the groove, the separation is very different. So now we have the major groove, which is deeply inside but barely accessible. It's almost closed. If we see comparison side by side, you see here the B helix with the grooves are very accessible. But here you have the sequence, which means the same sequence. So the sequence is the same, we just change the pucker here. And you see one helix, right now that's the other, A form, B form. Because then is how you control that. You see, going in the DNA, then you have, this is not very common. This is the most common packaging form. But in RNA, this is the common one because the ribose plays a key role. And the ribose is the key player, and the ribose stabilizes that helix. I will come to that. But with the same sequence, you can build both. Actually, you can change, you can adapt with the DNA, for example, go. And you see how here the major groove here is closed here. That's another view. RNA. Here is the major groove. You see it's very deep, but you see how narrow it is here. It's difficult to reach the bottom, the bases, to reach in terms of contacting the bases. And here the bDNA is the contrary. You see how open it is. That's where most of the transcription factors, for example, the regulation, they would bind here because that's the most accessible. That's where you can decode the bases also. the reading, because that's where you have most specificity. For RNA, in order to get there, you need either a special molecule, so very small, or you have to open distort. And here we come. That RNA is accessible to decoding, but in special structures. Not in the helical part, you have this part, which is the most accessible part. It's open, that's the so-called minor groove, which is very important in RNA. And see, in between, if you want to insert a molecule, you have to open. Here it goes very quickly, very easily, and here it would not go. It's a very simple concept. But, so the helix is the simplest way to show, but now you will see even in more complex structures that you observe with RNA, you can reach this part, but in special parts, not in regular helical stem. We'll see that. So RNA folding, you have two principles. Stacking of the bases, that provides energy, stability, and regularity. And base-base hydrogen bond. And these two are key for RNA structure architecture, but they are also key for helical stems. But with RNA, as you will see, we can do more. The first example of this complexity came from the structure of the transfer RNA. And here you have the sequence, but not in the linear form as you have usually, is the clover leaf. And the clover leaf itself, the finding of that clover leaf was an achievement. And because you could see that you had internal loops going from here to here, internal loops, stems, and this was correctly predicted. By correctly, I mean these interactions here could be seen once we could have the three-dimensional structure experimentally determined. And the interaction between these and these is not obvious. Having that, it took some time. Actually, it took the crystal structure determination in order to find a correlation between these two. But if you look now on this one, you will see how you can go from here to here. And you can recognize the part I will show you. You go from these, 2D and 3D. Here you have the helical part, two helices, and the three-dimensional part includes the three-dimensional contacts, which are essential. For example, this part and this part are contacting each other. That was not obvious at all. You know, when you had that, here you had the connections. Between that and that, nobody knew what was happening. It seemed trivial today. And here is the reversion with the name of what is classic now, the refolding. And there are special features on that transfer RNA. These structures, not only because the molecule is important by itself, which is already sufficient, it's an essential molecule in life, but in terms of structural information, going from what I showed you about the helices, where you have sugar poker and bases. Now we have the specificity of RNA, which is a 3D structure, which has, I mean, if you look at that from large, doesn't resemble an helix, just a classical helix. Of course, if you go there, you will recognize the helix. But here, that doesn't look like an helix. And then you have tertiary interactions. So you have more than just base pair. You have what was discovered there, 3 bases interacting, for example. And that makes specific contact. This is RNA. And you have long distance interaction coming. You have loops. And what I told you before about the accessibility of the stem, of the loop. You know, if you want to go here, the groove, you have the problem that I presented to you first, is the narrow aspect of the access to the groove. So you cannot. Here you cannot. I mean, although the picture is not so suggestive, you have the same problem. It's closed. But here at the end, the loop, at the end, you have the end effect then you have an opening, accessibility, from this side, but also from the other side, which gives, let's say, the tertiary structure of the RNA has to be seen on its own. And it can be, depending on where the critical point you want to reach with a given protein, you can or you cannot. And of course, this is a crucial part to be recognized, but also for control and regulation, you can have molecules here that control the conformation of that. And then here is the same. These parts are, every part of this molecule can be accessed by different proteins, or even by a given protein by different part of this protein. And then you can push and make either recognition or modify slightly. And you will see an extreme example of that with the ribosome, where the complexity is at maximum, where you have RNA doing a lot of things, but also playing with these molecules, just pushing the right motion and adapting. And so the sensitivity becomes very important. See? And everything goes back to very, very basic principle, the Parker basis arrangement, the tertiary. And you see here, that's a 3D in terms of accessibility. That's a very old picture. And here, for example, you see the EFTU. EFTU recognizes this part, elongation factor, this part. CDTs, we'll see later another, recognize this part and others, depending on the degrees of complexity. And other factors will recognize. Some of them will recognize the anti-common part, and because several molecules. And of course, for other RNA, you have the same properties, that according to the functional aspect that you want to emphasize, either the recognition or the activity, you want to change the conformation, recognize position, then you want to play with the molecule and adapt it. Because RNA is there as a backbone and is also adaptable. And more, it's more than RNA, you will see more complex structure along these studies. That's an example of a riboswitch. And this is another. We have seen the transfer RNA molecule. Now this is another sequence, SAM2. And you see the structure is not more recent. The structure presented to you first was the 80s, 74 the first, 1980 the second transfer RNA. It was pretty slow. Now we are talking about 20, 30 years later, and there are much more. And you see, again, the secondary structure is more complex, and the tertiary structure takes advantage, but you have a semi-helical stem. But in details, you will see that this part, which is interacting with this part, is coming from another part. So the complexity of the structure is something which is specific to RNA. What is the question that I was not responsible for the title of the introduction. Jim, thanks, Jim. I don't know if I follow what you wanted, but this is what is special about RNA, what RNA can do, special structures, special adaptation, and for specific purpose. But this structure also is not random. It's not that one and 100 other possibilities. It has, every sequence has a preferred unique conformer or an alternative of two. That's also very important. The structure is embedded in the sequence, right? And for me. And I think I will stop because I had a few slides just to show. This is a contribution from Eric Westhoff, who is giving the talk, a lecture on nucleic acid research at our university. I borrowed some slides for introduction from him, and he's well known for the ontology. And he wrote several papers in nucleic acid research that I recommend to you. I have a reference, and he emphasized the classification are due to the different edges of the bases which are recognized. Here you have the Hoogsteen base pair, Watson-Crick, and the sugar edge. And according to this, each of these parts can be recognized. So if you can combine that, and that makes a complexity of the structure. For the pyrimidines, you have the three, but you see how different they are. Here's the Watson-Crick remains in its place, but look at this one and this one. You see how different they are? And the glycosidic, also the angle they form is different compared to the backbone. And so all that gives you 3 edges, and 3 edges carry various edge bonding. So altogether, you have 12 basic families. You go back to the paper, so you can classify the RNA in basic ways. And that gives you a glimpse on the complexity of these structures. This is based on available structures. By the way, the structure of the ribosome and the intron, the first structure, the model was done by Michel and Westhoff, and before it was the three-dimensional structure confirmed the prediction, but the model came out of model building, and this is something quite an achievement that was done, and that is possible with RNA, and it's actually I would say we can do more with RNA than with protein in terms of predicting structure right now for that. But just the information I give you to give you, maybe not everybody knows that this model of intron, to build it, they use the only available information at that time, the transfer RNA structure. So they just cut the transfer RNA in small modules, which were typical examples of this type of interactions. As building block, they had the interactions by sequence comparison, of course. That's also another very important tool. I mean, I'm sure you'll come back to that in order to find what is conserved, what is not, what is key. That defines the interaction by taking these modules, and the only modules available were from the transfer RNA structure. They build a complex structure. When you look at the two structures, they do not look alike. I mean, they are totally different, but the building blocks are common. Now we have much more building blocks because we have much more experimental data, so we can build more complex structures. But this is just to illustrate to you how these things are chemically stabilized, chemistry behind, and the three-dimensional structure is not something just waving around according to the buffer and the ion and other things. It's the information content is three-dimensional. And that is what you speak. I didn't speak about catalytic properties and other things. There will be a lot of talks. That was just a glimpse to give you an idea. So RNA is indeed special, I believe it. Is important that you didn't have to listen to my talk to know that, but I confirm. And and there's a, sorry, I want to just thanks Eric who provided access to the document of his course, because most of the document, although they are publicly available for everybody, but just, you know, for me it was easier because I had a notice to prepare that course on a short time scale. Okay, thank you very much for listening to me.